Hello, everyone, and welcome back to Lady Scientist Podcast. I'm your host, Jocelyn Pearl, and today we have another great interview in store for you guys. I chat with Dr. Kiana Aran. She's an associate professor at the Keck Graduate Institute in California, and she's also the inventor of the CRISPR-Chip technology. We get into the story behind that idea and more in this episode. Before we jump in, if you've been enjoying our content so far, please click subscribe and leave us a comment or a review. It really helps us. I'd also like to give a shout out to our sponsor, Kendall Investor Relations. Thank you so much for listening to this independent podcast. I hope you enjoy this episode. Today, it's my privilege to welcome Dr. Kiana Aran. She's an associate professor at the Keck Graduate Institute, and she's also the CSO and co-founder of Cardea Bio. Kiana also was recently awarded Nature's Women in Science Award for 2021. Congrats, Kiana. And we're really excited to have you on Lady Scientist Podcast.
Thank you very much for having me.
So I want to jump in with all of the news that has come out around this award and some of your announcements. Can you tell us a little bit about what the award process was like for you?
Yes, absolutely. First of all, thank you so much for having me in this podcast. It's a pleasure. And the Nature Research Award was an award that basically recognized women in their early careers and their contribution to science. And I was, so apparently there were about 180 candidates, and 6 people were shortlisted, and I was super excited to be shortlisted. And for about a month, we had a meeting together, and we had some sort of leadership training and a chat room where we could chat about our challenges, which was really great. And just to meet those wonderful women, it was an amazing win for me. And on the day of announcement, I actually was not expecting that. And I literally did my speech talk prepared like 5 minutes before the ceremony, because I wasn't expecting it. So yes, so it was a sort of a surprise for me, and I was super happy, and I'm really excited to be the recipient of that award. It means a lot to me because I worked really hard to get where I am, and it was fascinating and exciting to me to feel that, the science that I was really passionate about was also resonating with others. So that was really good.
Wow, that's amazing. You mentioned in another interview about this group chat that you guys had as the nominees. Can you talk about maybe some of the leadership training that you guys did, and if you think that anything will come out of making those connections with these other women leaders in this field?
Absolutely. So in that platform where we were able to chat with each other, we're also provided with training from other women, other leaders, and some of the challenges they had. How did they create the brand for themselves? How did they promote themselves? How did they, deal, in general, how do they deal with conflict? And really, important topics which, as scientists, as leaders, we encounter on a daily basis, and ensuring that we really, take those experiences and really, react to them in a way that a leader would react, not, because as scientists, sometimes, we're so passionate about what we do, and we're so direct, and we're not very humble, I would say. And sometimes when you become leaders, you really have to tune some of those characters to become a better leader, and not that, being direct or being scientifically oriented, where we really have to see, hypothesis result, experiment results. Sometimes really just looking at it from a different perspective is really important to become a better leader. And it was nice. It was nice to actually be able to interact with those women and have those opportunities to hear their stories and how they got there, how they dealt with their issues and their problems. So I found it really interesting, and we still continue to be members of that platform. And I already created, connections with some of those women. I'm actually going to Berkeley next week, and I have dinner plans with the runner-up because she was from Berkeley. So we're planning to talk to see if there is any project that we can collaborate. And also, I wanted to get them involved in the nonprofit organization that I established with the award money that I got from Nature. And I wanted to get, all of the shortlisted women involved in that journey with me as well. So had multiple meetings, and I'm hoping to meet the rest of the team in person as well.
That's a great segue, because I wanted to ask you next about the new platform that you're developing and what you're doing with this prize money. So maybe you can.
Absolutely. So I'm a scientist. I'm also an entrepreneur. I always had the excitement of just taking my technology to next level, to a product. Throughout my journey, I would encounter a lot of things that I wasn't actually paying attention to. For example, I'll give you an example. This question of how does it feel to work in a male-dominated field? I have received this question so many times, and it sort of carries such a negative, energy for me, because it feels like you're putting everything aside and you're just focusing on me being a woman and working in a field that is, mostly male dominated, and you're making men look like really not human, like monster. So I just don't connect with that. And I think it really shifts our focus to limitation mostly than, what is possible, what is in front of you, how can I take advantage of that, how can I actually become a leader? Because if you constantly tell women it's so hard to work in a male-dominated field, it's so difficult, how did you do it? So you're making it even more difficult than it sounds like, right? So the next person who wants to take your path, they already, entering this journey with the feeling that it's going to be very difficult to work with men. It's going to be very difficult to work in a male-dominated field. It's not. It's not if you're true to your, to what you have achieved. It's true to your capability, and I agree that there are things that needs to be fixed, but, just constantly focusing on the root of the problem without basically stepping in, creating more opportunities, creating more role models so other people can actually look into. I think it's as important as fixing those roots, right? You can fix the roots when you have more females, when you have more women in those industries. So that was something that, I constantly talk with my co-founder and my colleagues at Cardea, when we were interviewed by journalists, and they would ask them, question, all technical question and business question. And when they get to me, that's the first question that they would ask. I'm like, no, not this one again, and then after winning this award, one of my colleagues told me, Kiana, maybe that's an opportunity for you to basically start something to act on, to just promote them. And then they have been helping me, actually, all my co-founder and Cardea, staff and talents have been all helping me to actually create this platform. So this platform is not focused on women. It's mostly focusing on how can we actually, promote roles in leadership for women. And the reason that, we have to fix the women issue in those roles to begin with is that there's so much focus on drawing more women into STEM field, and we have been doing a great job in advocating for it, in getting young girls excited about becoming a scientist, being involved in STEM. And if you look at universities, 50% or more of graduate degrees are awarded to women. But then when you look at the end of the funnel, when all of these resources have been invested in educating these women, you don't see many of them in leadership role. And really to make science impactful, we have to, collaborate. We have to bring all of these great minds together to be able to solve humankind problem. So to unlock the power of education, for all of these resources that goes into training women, we need to bring them more into influential roles. And how do we do that? We create, we bring more women in those roles. And the way that we can do that is bring women who have potential to become successful in those roles, and provide them with tools and necessary training so they're ready to take those roles. And also provide a platform when, for example, a firm or an industry is looking for a women leader, is looking for a woman board member, they don't just randomly pick someone and hope that it's a good selection, but they can go and have a platform where they can look at the experiences, the scientific background of these women, and they can at least see that these women, they didn't have any experience, they have not worked in the similar environment, but they have had some sort of a training, so they have a higher chance of being successful in those roles. And this way we can bring more women into the industry, which can then attract more women to those roles and create more role models. And I called it the nebula because the nebula is where stars are formed. And really, the force of gravity causes the stars, to form if they can overcome the outward force of limitation that is in front of them. So this is the goal: bring more women, create more stars, and so the rest of the women have role models in leadership roles. And this way, I think we can really, create balance, unlock the power of education by really utilizing all of these resources that is going into training these women in STEM. Because otherwise, just putting more women at the beginning of the funnel and not, which does not lead to any influential role. It doesn't solve the problem.
Yeah, I love that concept. And I just think it's a great way to build on this personal ethos you have of focusing on possibilities and not limitations. And in the near term, what's the ask? Is there going to be some kind of application for women interested in participating in this platform, or what does it look like?
So everything happened so fast. So basically we did everything within a week, right? And we're still establishing this thing. But as I mentioned, to begin with, the membership is very particular because we want to bring, women in science who have higher chance of becoming successful leader in STEM, in science. So we have to have some sort of a selection criteria for the initial members, so, and as we expand, we're really thinking of how do we expand it to others. But to begin with, there are some sort of a criteria. I don't want to spell them out because we're still, speaking with an expert, and I am planning to speak with Nature and really, brainstorm together to see what is the best sort of this selection for the first, 50 members that will be offered with training classes and tools, and how do we actually expand on that will come after. But the selection criteria is mostly going to be science-based.
Awesome. Well, we'll make sure to link that as well for this episode so people can go find it. And I agree, everything's happened really quickly. I was just prepping for this interview and I couldn't believe how much I needed to catch up on. So you've been very busy. I commend you on all of these new developments.
Thank you.
So I want to skip back a little bit in time and talk about the story of CRISPR-Chip, which, by the way, it's such a fascinating technology, and the idea of, marrying biomedical, reagents with electrical reagents is really novel to me in my brain. And so I love to just learn about the story of this technology and kind of how you came up with it and what that process was like working on it and developing it.
Absolutely. So being in Berkeley, of course, everything is about CRISPR. It's been about CRISPR for the last, 7-8 years. So being a postdoc in Berkeley, I was also fascinated about CRISPR. And I remember one day, it was a Saturday morning, and I was going to my exercise class in the morning, and in my mind, I was thinking about, deactivated CRISPR and how CRISPR is being used for imaging application and all of that. And all of a sudden, I stood by the Pete's Coffee in Berkeley, the first Pete's Coffee. A lot of people who are resident of Berkeley, they know that coffee shop. I stood by the coffee shop, and in my mind, I'm like, what if I could capture the function of CRISPR? Because CRISPR is this wonderful enzyme that can unzip the double-stranded DNA, find its target sequence with the help of a guide RNA molecule that can be programmed. And before we can do anything, it's searching. It's a search engine that can search a sequence of interest, and we program it to search for that sequence. What if we could just watch the enzyme as it does what it is good at? It's an enzyme created by nature over billions of years. It's a perfect enzyme. No human can create such an enzyme. And I was working on using transistors and modern electronic for the majority of my previous work. And transistors are very sensitive to charged molecule. So if you actually put a charged molecule on top of a transistor, you can sense it, because the majority of our protein, enzymes, and everything in our body has a charge, has an electrical charge in it. So I thought, okay, if I decorated, if I anchored a CRISPR molecule to a surface of a nanotransistors and watched it basically as it interacted with its target sequence, and once it binds to its target sequence, if I could actually see that signal, then we can solve a major problem, which is amplification. Because right now, the way we actually detect a sequence, we chop the DNA, we chop the genome, then we add some primers and we amplify the sequence of interest. Then we add some label to it, and then we can detect it with some optical instrument. Basically, as my colleague says always, is like we run over the sample by bus before we can detect it. What if we use biology as technology? And if this biological molecule can really do the search function for us, can we anchor it to modern electronics and watch it as it does the search? So that was the whole idea. And I was standing there for like 7-8 minutes designing the whole experiment in my head. The whole time during my workout class, I was designing experiment, in my head. I can vividly remember that day. And I could not, I called a few friends on a Sunday, and I'm like, who can help me with this project? And Monday we were at it. We were starting to working with the project. And at the time, I had never had my hand on a CRISPR molecule. I had never done a CRISPR experiment. It was just an idea. And as a biomedical engineer who was, trained on electrical engineering, biomedical engineering, bioengineering, have done chemistry, have done biology, I was not scared of, doing things or mixing things together, which I think it's a really a benefit of having that, sort of background, because if you told a biologist to put an enzyme with an electronic system, they would freak out, right? But for me, nothing was impossible. I would just mix sciences together, and it either works or I would fail. So I think that was the beginning of that journey, which then led to multiple different collaboration with different scientists, with industry, which then really led to me starting a company, then merging two companies. And it was the idea, I think, first, but beyond that was being brave enough to reach out, not being scared of sharing my ideas with others, and asking for help, and really bring great collaborators to help me take the idea to the next level. And today I have learned so much about CRISPR, and I'm fascinated more than ever about not only CRISPR, but also now other biological molecules and how I can capture their function. So it has become a theme in my lab where, can we use biology as technology? Can we look around to see what else is available for us to actually take advantage and how we can unlock those resources for human health?
So you mentioned this first week planning these experiments during your class. About how far down the road were you collecting data that made you think, okay, I think this is working?
So to be honest with you, the working part was within the first 5-6 months. We were getting very, very promising results. Um, uh, the major challenge was the electronic, because we used nano transistors, and I was fabricating those transistors myself. And when I became a professor, it was also difficult to go back and forth to Berkeley and fabricate those transistors. And that was a major limitation. And then after I found my collaborators, who are today my colleagues at Cardea, and they were actually making commercial scale transistors, and we were able to move our platform to their platform, or technology on their platform. It was really, everything moved significantly faster in terms of clinical testing and all that. And I realized that this had a really great potential to become a product. This was after a trip to San Francisco, where I heard about another company and their ability to fundraise. And then I decided to reach out to a network of people first who I have made during my first 2 years in Berkeley, where I started another company for another invention that I had. And I failed in starting a company. I just I did not have the experiences, or I did not know how to build the right team. But I built a big network. So I reached out to them, and then everything basically connected and helped me to form the second company, which today has merged to become a bigger company, which is Cardea.
And out of curiosity, was there a particular genetic mutation that you used as a positive control in those early experiments? Or I'm just actually curious about the process there a little bit.
Yes, absolutely. So to start with, we just used, I was collaborating with one of the faculties in Berkeley, and they had a model. They were using CRISPR, a model for those were HEC-BFP cells. I don't know, you probably are familiar with that. It's a blue fluorescence protein that is engineered into the cells. And they have designed successful guide RNA and complex to detect that gene. So everything was already, we started with a sample that was tested multiple times to work in vivo, and it was a very robust sample. So this is how we started, as a positive control and to make sure that the platform worked. Later on, we moved on to clinical sample, where we tested it against Duchenne muscular dystrophy, and we were comparing it with healthy genomic sample. And then later on, within 2-3 years, we expanded the technology to detect single point mutations in sickle cell disease and ALS.
Amazing. And what's the long-term vision for this product? Is it to be in hospitals and clinics around the world?
Yeah. So when I first started thinking about the technology, for me, it was the versatility of the CRISPR itself, where you could really program it to detect anything. That's one. Two is the potential, expansion of having a chip which had 1,000 transistors on, and each of those transistors could be potentially, we could potentially decorate them with a specific CRISPR molecule to detect a specific region. So when I envisioned this, I imagined a chip that could detect, hundreds of genetic diseases at the same time. But this was the initial thinking. But then as we moved to the industry and looking at, talking with customers and really listening to them and what was the need for the market, we really found other applications which were, which we could get to faster than having a, chip that could detect hundreds of genetic mutations at the same time, which we didn't even know that those were needed. For example, with our platform, you can monitor you can monitor and quantify RNA binding to proteins. And that was, we could, when we were functionalizing our chip, we would monitor when we actually added our guide RNA to interact with the Cas protein to form the complex. And that data itself was so valuable, and we realized that there was no instrument out there to actually quantify that. And it's not only important for CRISPR design, but it's important for a lot of other therapeutics applications that are working on RNA binding proteins. So, basically people who are working in the field of epitranscriptomics, they know the importance of, RNA binding protein. And so we're doing some RNA binding protein. We're working on using the same exact platform for quality control application for CRISPR. Because a lot of time, designing the right complex is super important. The same exact platform that we developed with the same exact, methodology of, working methodology can be used for monitoring the stability of the complex in different environmental conditions. If you have a cell that is cancerous or if it's a cell that is healthy, how do you expect your complex to function? Or, when you're doing editing in some sort of cells, how much editing you actually have achieved, and you can monitor before and after, editing, and to see what was the percentage of your editing in cell type. So these were the applications we did not think about, that are, that arise, later on, but it led to a different company that we did a spin-off from Cardea Bio called CRISPR-QC, which we're actually doing just for CRISPR-QC. Also, what we did not think that, there's a lot of need in agriculture for having a handheld system to monitor the presence or absence of, some sort of genes or transgenes, and our platform could offer that. So, as you talk with customers, as you talk with people, each of them help you to realize where can your product be more useful. Yeah. And it was a great lesson for me too, that in order to develop technologies in my lab that are meaningful to society, I really have to talk with people and industry to see what the needs are. And it has really shaped the way that I actually do my research in my academic lab as well.
Wow. It sounds like it was one of those ideas that, once you built the initial proof of concept or minimum viable product, it just kind of expanded from there, and there was really a ton of different directions that you could go with it.
Absolutely.
Just to get a sense, timing wise, what's the comparison as far as a readout from this type of device versus something like traditional PCR, like you were talking about earlier?
Yes, absolutely. So remember that our platform does not require any amplification, right? So you basically put the whole genome in and you let CRISPR do what it's good at, right? You basically let the biology take care of it. So that itself cuts a lot of time. And the rest is basically sample type. So for some of the samples, we needed about 20 minutes. For some of the sample, 5 minutes was enough, So the average is about from the time that you have your chip functionalized and ready, and you put the genome on to where you have the readout, it's normally 15 minutes.
Wow, that's incredible. And really, could change how quickly a lot of, science and molecular biology is performed.
Exactly. And then we're reading it, right? We're reading biology class. We're reading the CRISPR enzyme do the work for us. So
I want to get into the nuts and bolts of what your lab is like and how you run a lab that bridges these two different fields, really. What's the setup like? What kind of people do you recruit, and what are their backgrounds?
Really great question, and a very difficult question. So in my lab, we also work a lot on aging. So we do a lot of diagnostic, but we also use our tools to discover, some biomarkers for aging too. So the lab is basically divided to two parts, where one part is really sensor development and the next generation of sensors using modern electronic, and the other part of the lab basically utilizes the tools that I've already developed throughout my years of research to identify biomarkers for aging, and we collaborate extensively on all of those projects. Who do I recruit for the lab? It's a great question, because it's of course a combination of, hard skills and expertise that you have. And as I am through my journey, as I am learning to become a better leader, I'm also paying more attention to soft skill and learning to really identify people who have the same motivational factors, or at least understand their motivational factor to make sure how do we actually put the team to work together. It's a very, very collaborative work environment. Projects are team-based. There is not a single project that a single individual is working on. There is really teams that work together based on their expertise. And we really foster collaboration and innovative thinking. And I think we live in an era that all sciences are required to have a successful project and a successful product. And so in my lab, we have chemists, biologists, protein engineers, electrical engineers, physicists, and just name it. And we also have a lot of undergrad. We have high school students who work with us in the summer. And I love working with undergrads and high school students because their mind is so open, and they're not scared of trying things. And sometimes it's really nice to see that. And, they really work with senior scientists, and I just enjoy seeing this collaborative environment. And I learn on a daily basis from my people, because I don't have time to actually learn about all of those topics. And with experts that I work with, it's a constant, learning for me, which what makes my job really exciting.
You mentioned working on soft skills and thinking about how you lead. Has there been any learnings from this past year for you that were really eye-opening as far as changing how you lead others?
Absolutely. So I myself come from a culture that is very different. And because we had, some limitations, sometimes we were very direct in our in the way we acted. And throughout this learning, I have identified some of the things that I had to basically tune. Being kind, it's good, but it's not always good, right? Being assertive, it's not good, but it's not always bad. How do you actually tune these things and make it an individualized based on the people and group of people that you work with to make a win-win situation? Because you want people to really enjoy where they are. I'm enjoying what I'm doing, but I really want my talents and people who work with me to have the same feeling. And it's been a training for me to just try to see things from people's perspective, and just zoom out a little bit and see what is it that we can find in common together so we can create a better work environment. And in science, doing science in the US, that means you have to really be able to work with diverse group of people from different culture, different background. And that means that you have to study their culture. You have to make sure that, if you're talking with someone, and if things are said, these are not personal, these are coming from cultural background. And understanding and respecting each other's culture and just enjoying that, it's I think has been a major learning lesson for me, and has helped me a lot. Like sometimes I tell my colleagues, how did you work with me 2 years ago? And because I feel, sometimes I'm super excited and super passionate about doing things that, it might come as too assertive, and I have to tune that a little bit. So it's been great. It's been, I think, when you, as a scientist, when I look at it as a science, and I'm like, wow, this is really powerful stuff, and creating that work environment, it's been amazing. And it's the funny thing is that it allows you to not have any negative energy because of some of those conflict that might arise from different cultures, and that not having that negative energy will result in having more time, more positive energy to do better things that are more important, and they can really impact life. So it's lessons learned on a daily basis, tuning on a daily basis. And as a leader, I also have to make sure that the team are playing well with each other. And to do that, you have to teach what you learn. And I think through teaching of this leadership skill, you also hold yourself to higher standard, that you have to actually do what you learn, because then it's not learning. You have to, you have to really do what you know, because then it's not knowing, right? So it puts a bit pressure, but it's also, I think, it's a nice thing, and having that higher bar, it's really great.
I love that. Um, yeah, I think it's such a valuable skill that we all, have to develop over time and learn how to interact and be the best selves we can be for the people that we work with or otherwise. I wanted to ask, you recently participated in this World CRISPR Day. There was a panel of women leaders in CRISPR, which I thought was such a great event. Several women contributed to it and shared their experiences, and there was a lot of enthusiasm in the chat for that session, as far as women just feeling like they were hearing things that they could relate to. I thought it was really powerful. So I was just wondering if you could talk about that experience and any takeaways you had from that event.
Yes, it was an amazing event. I also, it was interesting for me to actually learn about other panelists, to see what sort of perspective they had, right? And when you look at, or when you listen to those conversation, each of us had our own perspective, right? So it's really not a single formula that worked for any one of us. It was very different. It was, the motivational factors were different, and the way we saw it was really, really different. And what does that tell you? That tells you there is not, just focusing on problems and roots. It's not going to solve anything, because each of us have different problems, different root, different perspective. But what it's important is for other younger, scientists and younger women, to see more women in the field, despite all of this, their different challenges, ending up in those leadership roles. I think that was a powerful message, because that tells you each of us have challenges in life. They either societal, economical, or other sort of challenges, and we each face different challenges. But that does not mean that they can stop us or limit us in achieving our dreams. Some of us could have challenges because we are young mothers. Some of us, we can have challenges because we migrated, or we are immigrants. Some of us might have challenges because of other sort of limitations. But what's important is that all of us ended up in a leadership role. So don't be scared. Take the opportunities, and take it one step at a time. We don't have to be perfect, but that does not mean that we have to stop our progress because we want to be perfect. We should just take it as it comes.
Yeah, I really commend you on your positive nature and your attitude around building and not focusing on, the negatives. And I just think it's such an important kind of state of mind to put out in the world. And I really have enjoyed learning that about you over the course of these various events that you've been participating in and sharing your experiences through. And I think this conversation will add to that, ethos that you have about building and really creating and not limiting ourselves, right?
Yeah,
So.
I think all humans are great. We all have so much. We have extraordinary capability, and we shouldn't let our limitation define us. We should just focus on those possibilities, the extraordinary, capabilities that life offers us, and really take advantage of that to shape our future.
Absolutely. I think that's a great note for us to wrap up on. And I just want to be cognizant of your time and thank you for taking the time out of your busy schedule to share all of this with us. And I can't wait to get the episode out and just share it with more people. So
Thank you so much, and thank you for doing what you're doing. It's amazing to meet you as well.
yeah, you too. Thank you so much.
Thank you. Have a great day.
You too. That wraps up my interview with Dr. Kiana Aran. Kiana, thank you for taking the time to share your experiences with us. I'm so incredibly inspired by your passion for science, your inventions, and your drive to build and create and embrace possibility over limitation. I think it's such a great personal ethos that you put out into the world. If you enjoyed this interview, please click subscribe, leave us a comment or a review. I'd love to hear from you all. And let me know if there's anyone you'd love to see interviewed on this show. I'm always open to suggestions. If you enjoyed this episode, I recommend you check out the interview I did with Celine Hollywa, the CEO of Loyal, or the interview I did with Lindsay Pino, the co-founder of Talis Bio, both great personalities and great people to learn from. Thank you so much for listening and supporting this independent podcast. We'll see you next time.